So in this video, I'm going to tell you a little bit about how to use some of the basic search tools for dengue virus. So after you navigate to the dengue virus page, you can click on Browse All Search Types, and this will give you a list of the different data that's available for you to look through on dengue virus. If you'd like to search for specific sequences, you can click on Genome Data, and this will take you to a page where you can select the different search criteria that you might be interested in. If you look at the top of the page, you can see this number in red, which indicates how many sequences are available, and this changes in real time as you select the different search criteria. And these different search criteria can include the virus types, so you can search through it by the taxonomic tree, the collection year of the viruses, the geographic grouping, which includes both continents and individual countries. You can also select by host. And what I really like about ViPR is that it also lets you only select for complete genomes. So we have about 32,000 sequences, but if you select complete genomes, then this is reduced to only several thousand. We have about 6,098 complete genomes. Another special feature that's available for dengue viruses is that there are several datasets in ViPR for which clinical and sample metadata are available for. So underneath Core Project over here, you can select the project identifier, the grant agency, the sample provider, principal investigator, if you have a specific dengue sequencing virus project that you're interested in. So if we close that and click on Core Sample, here you can find information relating to the host or specimen type that you're interested in. You can also select by host age. If you open up project-specific data, here you can find information on the clinical status of patients. For example, the disease stage, the disease course, whether they recovered or whether the patient died. You can also search for other measured attributes, such as hypertension, whether there was any hemorrhagic symptoms, shock symptoms. It's actually quite detailed. There's passage history on certain viruses. And you can also look through advanced options, which will give you a couple other things that you can do, including looking by month range, as well as the ability to remove genome sequences. And I really like this option to remove duplicate genomes because this discards any sequences that are identical to others, and this can be really useful for comparative sequence analyses such as tree building, or if you're just trying to catalog all the different types of sequence variation. So going back to the main search page, I should also mention that if you're interested in doing this the other way around, you just want to retrieve any sequences that have associated with them specific attributes of clinical metadata, you can also make your selections by clicking on human surveillance data records and selecting the appropriate criteria. And again, this gives you all of the, it shows you that we have about 3,000 samples which have clinical information associated with them. And this is very detailed, and if you look in advanced options, there's several other display fields here that are not listed that you can also choose to include if you're so interested in doing so. So going back to the search tools page, another thing I'd like to tell you about is our immune epitope database. So if you click on immune epitopes, you can explore both experimentally determined as well as predicted immune epitopes. And right now there are just over 400 epitopes in the database for dengue virus. And these include epitopes that are recognized by B cells, T cells, and the major histocompatibility complexes. Some interesting options that you might want to test out or play around with are restricting by host, for example, humans or mice, or humanized, let's say, for example, mice. And if you look under advanced options, This also gives you a couple of nice little extras, such as being able to search in a certain amino acid range, or you can select by MHC class that you're interested in, or if there's a specific MHC allele that you're interested in, you can click on that, and this gives you a list of options, let's say HLA-2D class 1. And you just go ahead and click on search after you've selected your criteria, and you should get your results. And then one last thing I'd like to show you is our 3D protein structures database. And these are downloaded from PDB, the Protein Data Bank. And you can click here to browse or search for structure files downloaded from ProteinDB. So let's say, for example, you'd like to look at the structure of the envelope protein of dengue virus. You would click on that. Let's say you want to select all dengue viruses, and then you have the option to choose whether you want proteins only with experimentally determined epitopes, or proteins that have experimentally determined active sites, or that have specific sequence features. So right now there are about 10 structures in the database that match the envelope protein. And if I click on only proteins with experimentally determined epitopes, that gives me back 4. So I'm going to go ahead and click on search so you can see what the results look like. And the results will give you a table with the PDB ID and a brief description of the structure, the uniprot ID, the protein that you've obviously looked for, which is the envelope protein, the strain if this is known, the species of a virus if this is known, whether there's any subtype, any protein accession numbers that are associated with it, and the experimental method that was used to generate the structure, either NMR or X-ray diffraction, for example. So I'm going to go ahead and click on View Structure for this one, 3UAG, 3UAJ. And this takes me to a page where I can visualize the structure in the JSMOL software. And if you want to know a little bit more about it, you can just click on this link over here. And there are multiple ways in which you can view these models. Right now it's in the ball and stick conformation, and you can move it around and rotate it and turn it up and down in 3 dimensions. And you can also change how you visualize it, whether you want a space filling model, or if you're more comfortable, for example, looking at the primary structure in different colors. You can also look at the structure, the secondary structure, so the beta sheets and alpha helices. You can zoom in and out to specific regions that you're interested in. And if you look further down here, you have a couple of other options. You can highlight ligands in green or choose any other color that you'd like. So if I do that, it shows me a couple of different spots. And then if I take that off and I go down to highlight epitopes, this will show you the different epitopes that are found in this structure. So you can click on whether you want T-cell epitopes or MHC epitopes. And this gives you a list of what's available in our epitope database as well. And they can be highlighted directly on this protein. So I'm going to just go ahead and click on this one, and it'll show me in yellow or any other color I choose where these are. And you can save a video of this, you can save an image of it, and it's a really nice tool to just have for your publications, especially if you're looking at a sequence nucleotide polymorphism that might affect the structure or function of your protein. So that's it for my brief description of search functions. I encourage you to look through the other types of data that we have available on ViPR, and we have really diverse and large sets of highly curated data for you to explore, as well as multiple tools for you to perform your analysis with that have been specifically designed for these virus families. And finally, I'd like to note that you can always reach out to us for support by clicking here on the top right. Under support, you can report a problem if there are any glitches in the website, or if you're unable to do something, you can ask a question if you just want help. And we also provide web training for specific organizations or labs that reach out to us And we do this free of charge as part of our outreach budget. So please don't hesitate to reach out to us if you have any questions or want any help figuring out how to do something or asking about whether we have a specific kind of data on our website. Thank you, and I hope you found this useful.